So I think we will go ahead and get started. I'm seeing a really great group on the line. Hello, and thank you for joining our webinar today, Clinical Trials, What Are They and Who and How Do They Help? This is hosted by the Epilepsy Foundation in partnership with both Jazz Pharmaceuticals and the Rare Epilepsy Network. I'm Dr. Kathleen Farrell, the Vice President for Public Health and Outcomes at EF, and I'm joined today by two excellent presenters, Dr. Jackie French, Chief Medical and Innovation Officer at the Epilepsy Foundation. She is an epileptologist and professor of neurology at NYU, and Victoria Artiega of the SYNGAP Research Fund and the Hispanic Society for Rare Diseases. Clinical trials, as you know, are an important step in the development of new therapies for epilepsy. And this session is dedicated to addressing why and how they are conducted and what people with epilepsy or people with loved ones who have epilepsy should know if they want to consider participating in a clinical trial. Your questions today are very welcome and encouraged, and we'd ask that you please use the Q&A capability in Zoom. You should see that in your Zoom menu.
Hello, everybody. It's just wonderful to see everybody from all corners of the world today to talk about clinical trials and a little bit about them and whether you should consider being enrolled in one. So let's have the next slide. So let's start off talking a little more broadly about clinical research. Clinical research is the study of health and illness in people to learn how to prevent, diagnose, and treat illness. And really, clinical research is subdivided into observational studies where we just try and see why things happen in a certain way. There's also a basic research that doesn't involve human beings. And then there are clinical trials, which we're going to be talking much more about today. Clinical trials are a way for researchers to look for new or improved ways to treat, detect, or prevent a disease. That would be really nice. And the treatments can include new drugs, surgical procedures, medical devices, and they are ways to say, is it better to do it in this way or is it better to do it in that way? And most of the trials that we'll be talking about today are to demonstrate whether new therapies are effective or not, and those are called clinical trials. And they are often randomized, and we'll talk about what randomization means. But before we get off of observational studies, I just want to say that you may find that the place that you're receiving your care or your loved one is receiving their care may invite you to be in an observational study. That will help us understand better who gets better and why, or why things happen certain ways in people with epilepsy. And I encourage you to be part of those trials. It's a way to give back to the community because that information then can be shared with everyone and can help everyone. And the thing about observational research is that it will not change the way your physician treats you. So that will be the same, and it'll just be somebody looking on to see what the outcome is. Next slide. So why do we do clinical trials? They're probably, you're aware that many years ago, people used to be able to sell anything that they wanted and say that it was a medicine, whether it was snake oil or something that was unlikely to work or even could harm people. And at some point, actually in 1938, an act was passed by Congress to say the US government needs to be sort of on top of determining whether new therapies are safe and whether they will actually benefit anybody. So this is a charge from the government, and it is overseen by a government organization called the Food and Drug Administration. So the Food and Drug Administration, or FDA, you've probably heard of them, is charged with ensuring that drugs are not just safe, but also effective. And that also is true for different kinds of therapies before they're put on the pharmacy shelves, or for example, before a device is marketed and insurance will pay for it. Very importantly, they also are responsible for that very long piece of paper that you pull out of your medication that tells you everything about the drug, everything that could possibly go wrong, any conditions that you need to be careful if you have them. And so in order to get all of that information that goes into the label, they have to make sure that many, many people have been on that drug and that we really understand the safety profile of it and the risks and benefits. And the FDA imposes very strict rules that govern the conduct of clinical trials and really tell us the path that we have to take when we are evaluating new therapies. Without clinical trials, the Food and Drug Administration cannot allow new therapies to be on the market. So it's really important that people step up and enroll in clinical trials, which in fact can also benefit them so that these new therapies can move forward and eventually can be available for everyone. Next slide. And by the way, for the person from the UK, this is true in Europe as well as in the US. It's only a difference of name. So in Europe, the European Medicines Agency, or EMA, is responsible. In the United States, it's the FDA. Now, what do I mean by a clinical trial? Let's go way back in time and talk in the 1600s about the very first clinical trial that was ever done. And it was done by a guy by the name of Dr. Lind. And at that time, many sailors were dying as they went across the ocean from a disease called scurvy. And nobody knew what caused it, and nobody knew how to prevent it. So Dr. Lind basically said, okay, I'm going to try and do this in a scientific way. And I am going to take 2 people and give them a quart of cider a day. 2 people got elixir of vitriol. I don't know what that is, but it doesn't sound good. 2 people got vinegar. 2 people got seawater. 2 people got a mixture of garlic, mustard, and spice. I hope they gave them like some chicken to put it on or something. And 2 people got oranges and lemons. Well, it turned out that scurvy is a disease that's caused by lack of vitamin C. So of course, oranges and lemons were in fact the treatment. And after 6 days, the soldiers that got oranges and lemons got up from where they were lying very ill and began to take care of all the other people. And that's how we learned that in fact it was vitamin C and oranges and lemons that cured this disease. Now, in this case, we didn't need very many people in a trial in order to determine that that was the outcome. Unfortunately, as I often say, we don't usually have something as good as oranges and lemons for scurvy. So we need many, many more people in order to determine whether a new therapy is effective. Next slide. So how do we find the medications that ultimately are going to go into trials? It's kind of a little bit looking for a needle in a haystack, because there are thousands or millions of chemical compounds that have effects, that could have effect on the brain, could have effect on different cell functions in the brain. And out of those, maybe hundreds are identified that we know a little bit more about, and they are put into animal models. So, animals are given epilepsy in various ways, and then these chemicals are put in to see whether they work in the animal models. And if they do, they then go through rigorous safety testing in animals before ultimately maybe only 1 or 2 of those compounds will move forward into human beings. But even when they move into human beings, you'll be happy to know that it doesn't go straight from the animal model to the person with the disease. In between, there is testing in very, very brave healthy volunteers. So hundreds of healthy volunteers that don't have the disease take the medication, first in very low doses, and then higher and higher and higher doses, and then first only a single dose, and then multiple doses, and then over multiple weeks. And that's so that we know that there is not a major safety issue going to happen. And then, and only then, will we ask people with the disease in question to be in trials, because we feel it's very important for those people not to be taking too great a risk with their health, because of course we know they already have issues with their health. We don't want to make things worse. So once the drug has safe passage through healthy volunteers, we then go into a new phase of testing, which we call phase 2 and 3. And those are the phases where people with the disease, and in this case, of course, people with epilepsy, will be in trials to determine whether that drug is safe and effective. And hundreds to thousands of volunteers with the medical condition will go through those trials in phase 2 and phase 3 before the FDA will say, yes, I see that this drug has been effective and now and is safe, and now we can move it into the public domain. Next slide. So one thing that people sort of get stuck on is that, yes, indeed, these trials are run by pharmaceutical companies, and a lot of people may sort of say, oh, evil pharmaceutical companies. And we've heard a lot, certainly in the last couple of years, about pharmaceutical companies and whether they, may always tell us the truth, or whether they're, making too much money. But, a lot of these drugs actually start in universities and research centers. The ideas for these drugs and why they might work. Somebody in a research lab may say, oh, I think that seizures may be associated with too much inflammation in the brain, and they may test that theory, and eventually they may come up with a molecule. It's interesting because I was just talking to a researcher today about this, but they would not have the capability to get that drug all the way through all of those trials and get it approved for use. And that is where the for-profit companies do have to come in and do all of that. They're very good at doing it. That's sort of their role and their job. There is just no way for a drug to go from a medical doctor or a researcher right to the clinic. So even if it comes from the National Institutes of Health or someplace else, it's going to end up with a company. And that's why we as physicians, as the Epilepsy Foundation, and you as consumers need to partner with those for-profit companies, the pharmaceutical companies, so that we can have the best trials that we possibly can. Next slide. So as I said, phase 2 and 3 is where we test whether the medication will be safe and effective. And we say phase 2 and phase 3 because usually you need 2 trials to determine whether the drug is effective. And these 2 trials are done compared to what we call a control group. And I'm going to talk about that in a second. And that control group is usually given what's called a placebo. And I'm going to talk about that in a second. So for a device trial, often we only need 1 trial, but for pharmaceuticals, we need 2 trials. Next slide. So what is a placebo and why do we use it? Unfortunately, clinical trials can't be just like clinical practice. I can't just go in a trial and say, Mrs. Smith, please take this drug, tell me how many seizures you had, and I'll tell you whether the drug worked. There's too much chance that events, whether it's improvement or worsening, can happen just by chance. And whether it's good or it's bad, it's going to be attributed to that intervention, right? I give, 10 Mrs. Smiths the drug, and, 3 people get better, and 1 person has a terrible adverse effect. And I don't know whether that would have happened whether they didn't take the drug. So therefore, we have to have a control group with people who don't get the intervention. So we want to know what would have happened to that group, to a group of people, if the intervention or that drug had not been given. So that's what we call the control group. And we compare the group where we give the intervention to the group where we don't. And if you don't have a control group, usually you'll overestimate how effective an intervention is. Next slide. So that's where placebo comes in. So we could just say, okay, all these people over here don't take the drug. All these people over there do take the drug. And what do you think is going to happen? Well, the people who are told, I'm not getting the drug, they're going to be very disappointed. And, they are likely not to get better. The people who are given the drug are going to be very excited about it. And they might actually get better just because they have an expectation that things are going to get better. So we really want things in the two groups to be exactly the same. So we give the other group what's called a placebo, and that is an inactive treatment. It may be a pill or a tablet or an injection or a device, and it looks exactly like the real medicine, except it doesn't contain the active medicine. So it looks the same, but it's just basically a non-effective sugar pill. We say sugar pill, but it doesn't really have sugar in it. I just want to reassure people, we don't tell you take away all the things that you're currently using and just use the new medicine or the placebo. We actually add either the new medicine or the placebo on top of what people are already taking. And that's because we certainly don't want people to get worse because they're in a trial. And taking away the medications that already may be partially helping them could be very, very problematic for making them worse. Next slide. So then, knowing what a control group is, knowing what a placebo is, the clinical trials typically have several periods or parts. So the first part is what we call the screening baseline. So that's where we figure out about how often seizures are happening, because we want to compare what happens after the intervention, whether it be a device or a drug. So there's maybe a month or two, you're just counting seizures in a seizure diary. And then at one point, you get randomized to either be in the control group or be in the active group. And the control group, again, is going to get the placebo added on. The active group is going to get the actual drug added on. And then we're going to count seizures in a diary again, usually for approximately 2 to 3 months. And then we're going to compare how did the people with the active drug do versus the people who just got the placebo. So the time when people are actually taking the drug or placebo, that's called the treatment period. Now you're going to think to yourself, well, why would I go into a trial if I could get the placebo? I'm never actually going to get the active drug, and I'm not going to have a chance to get better from the drug. Because of that, there is a follow-up period after that randomization period where people are in the different groups. And in the follow-up period, everybody usually gets the opportunity to have the treatment in what we call an open fashion. So no longer do we have a placebo that looks like drug and drug. We just have everybody taking the drug and seeing how it does. That is not the part that is of interest to the Food and Drug Administration, but obviously it's of great interest to the people who are in the trial. Now, not every trial has that period. So if you were to be offered a trial by your doctor or somebody who provides your medical care, it's very important to ask what happens after the randomized placebo-controlled period. Is there a period where everybody gets the drug? And if so, for how long? Next slide. So this is kind of what it looks like. People are taking anywhere between 1 and 4 anti-seizure medicines, otherwise known as anti-epileptic drugs, during the baseline period. Then usually they get randomized. And again, neither your doctor nor you would know if you were in a trial which arm you had been randomized to. And it is by a flip of a coin. We'll talk about that, I think, on the next slide. But deciding who goes into which group is done by the flip of a coin. So nobody knows who's in which group during the study. And usually there's a couple of doses of the drug, and then there's the placebo during the treatment period, and then there's the follow-up period that I talked about. As I said, people continue to take their prescribed medication throughout. during the baseline, during the treatment period, and during the follow-up period, then, if this new medication or device or whatever is working for you, then you can make changes in your other medications and perhaps reduce what you've been taking. Next slide. So, as I said before, volunteers for trials have to accept the possibility of being randomly assigned to either placebo or active study drug. In the example that I gave before, and again, this is an important thing for everybody to ask if they're offered a trial, there were 2 active doses and placebo. So that means if you were in that trial, there would be a 2-to-1 likelihood that you would be on an active drug rather than the placebo. But that's going to change from one study to another. So you should ask that question about what is the chances that I will get placebo. But again, remember that that placebo is only during the treatment period, which is 2 to 3 months. And then after that, people can go usually to the open label where they will get the drug. We think long and hard about, leaving people on placebo for as short as possible. But without some period where we have that control group, we would never be able to know if the drug is really working. And that is really important for the millions of people who are going to take that drug in the future. Next slide. Do we need more anti-seizure medicines? I'm here to say I certainly think that we do, and I'm sure a lot of you agree with me. There are a lot of people with newly diagnosed epilepsy who get on a medication and are completely seizure-free and do extremely well. Fortunately, about 60% of people, but about 40% of people don't respond to the anti-seizure medicines that we currently have. We call them therapy resistant, and that's far too many. So unfortunately, although we have been developing new drugs and the number of seizures that people have has been going down over time, but we still have about that 40% who are not completely seizure-free, and we're not going to stop developing drugs and devices until we get everybody seizure-free. And as I'm sure many of you also are aware, some of the new and old anti-seizure medicines are not very nice to take. They can make you dizzy, they can make you sleepy, they could give you issues when you're pregnant in terms of their safety. There are many things that we can improve upon, and that is another thing that we're certainly trying to do with new medications, is improve upon those things. Next slide. But that's not all. We're looking for new therapies that could possibly prevent epilepsy. Or, wouldn't it be great if instead of having to take a seizure medication for the rest of your life, you could take a medication that actually makes the epilepsy go away so that eventually you don't need medication anymore? That's the dream. That is an anti-epileptic drug as opposed to an anti-seizure medicine. We want drugs that address comorbidities such as, thinking problems and mood disorders and anxiety and other things that go along with epilepsy. And, we are trying to develop new therapies that address all of those things. And, recently we've had gene therapies and other novel treatments that give us really new hope that we can really make a substantial difference. So it's an exciting time for new therapies. And, of course, people who enroll in clinical trials may get an opportunity to try these new therapies, 5 years before anybody else gets to try them. Next slide. So without volunteers for clinical trials, no new medications or devices would be possible. So we really rely on people to volunteer for these trials. And again, the hope is that they also can benefit themselves. I have a gentleman that I take care of who volunteered for 10 different clinical trials. Some of them made him a little better, but they didn't make him all the way better. So he kept going into trial after trial after trial. And I'm happy to tell you, knocking wood here, that he now has been on his last clinical trial seizure-free for 2 years. So he finally found that magic bullet for him. Sometimes it's not the first time. Couldn't have been that bad to be in a trial since he he came back for more. And many new options are on the way, providing hope for everybody with uncontrolled seizures and side effects and other things that we want to improve on. And I am happy to take questions at the end.
Thanks, Jackie. There are some really great questions in the Q&A box. A couple in the area of, are people who are well controlled either by medication or otherwise, do they have an opportunity to participate in research studies or clinical trials?
So it would be a different kind of trial, right? We certainly don't want to cause seizures in people who are well controlled by, for example, taking them off their medication and seeing whether something else would work. But there are certain kinds of trials, maybe for, something else that's going on with them. Maybe they're well controlled with their seizures, but they have a mood disorder, for example, and would be a candidate for a trial for that. Or maybe there is a device that, helps them look at seizure risk in the future. So there's all kinds of trials, and everybody, should ask their doctor, is there, I just want to let you know, I'm willing to consider, you obviously don't have to say, I'm going to go in the next trial you offer me, because you have to know what that trial is, but I'm willing to consider. So if you want to present me with anything, please do.
Thank you. I'll give a couple more questions before we move to our next speaker, but encourage everyone to continue putting questions in the chat. Do natural history studies necessarily need to be done before a clinical trial, or does that vary?
No, natural history studies do not need to be done for most conditions. Right now, there are many natural history studies that are being done for rare epilepsies because we really don't know what happens to people with some of the rare epilepsies due to gene disorders over time. So, for example, what would happen if, and this is true for some rare epilepsies, seizures are very bad for a year or two, but then they naturally improve after that. You would have to know that that's going to happen, because obviously if you randomize people into trials at that time, everybody would get better. So you need to know what happens over the course of time and when it happens to be able to design your trial for the intervention. For focal epilepsy, which is, the epilepsy that's the most common, we have done so many trials, we know what the natural history is, and we don't need to do it again.
Thank you. And just one more, and we'll circle back to more questions at the end of the session, but what impact do you think genetic testing will have on potentially more targeted approaches in therapy development or clinical trial design?
Well, we certainly are learning. So the genetic epilepsies right now that are candidates for trials in general are what we call the monogenetic epilepsies. That means that a single gene is responsible for the condition. There are many people with epilepsy who have what we call polygenetic epilepsy, which means that there may be many genes that are contributing, and it's sort of like a pileup of, of things going wrong until suddenly epilepsy pops up. Those are a little harder to deal with. But with the monogenetic epilepsies, the more we learn about the single genes that cause epilepsy, the more we can go in and try and correct them very early before they cause the harm that they do. So, and then, on top of that, once we know, an example is Dravet syndrome, which some of you may have heard of, that is caused by a mutation in the SCN1A gene. We know for a fact that there are way, way, way more cases in America than we know about. And when we're trying to find the right therapy for people and put them first in the right trial, and then eventually when the drug goes on the market, give it, as a marketed drug, the more of those we know about, the better. So that's going to help us find candidates for trials and then ultimately help us target therapy.
Thank you very much. I'm going to stop my screen share, and Victoria, I'll ask you to please share your screen. And you can begin when you're ready.
Thank you so much, Jacqueline, for the excellent presentation, Caitlin, for your invitation, and the Epilepsy Foundation for for this invitation as well. I am Victoria Arteaga. I am a board member and Latin American director for SINGAP Research Fund. I'm also a co-founder of Hispanic Society of Rare Diseases. But the reason why I'm here today is because I want to share with you my daughter. She's Amelia. She's 7 years old. She was diagnosed with SINGAP1 at age 3. SINGAP1 is a genetic disorder caused by a variant on the SINGAP1 gene located at the chromosome 6. This leads to many neurological issues, just like epilepsy, intellectual disability, severe behaviors, autism spectrum disorders, and so on. When we got the diagnosis, it was really hard. It was a change of what we were hoping for the life of our daughter, well, the path of our life as a family. It took us probably a year to kind of like grieve through these new news on our lives. You know, finding her the right treatment for the seizures she was having, and at the worst moment, she was having up to 100 myoclonic and drop seizures. Finding she's now controlled with 3 medications. It took us a long time to do it. Also finding the right school and everything, but then even though my background is international business, I didn't understand anything about all the scientific papers that were there. Amelia was at the time the 200th patient in the whole world that was diagnosed with SINGAP1. So there were a lot of questions on what was the treatment going to be. It wasn't a treatment there. So what is going to help us improve her quality of life? So that's when we become more active as patients and later become patient advocates because we wanted at the time to be active participants on clinical research, which is very important. So initially with Amelia, we traveled to Edinburgh, to the University of Edinburgh, to the Patrick Wall Center to be part of the neuro-GD study, social responsiveness studies, sensory symptoms, syndromic autism and sleep studies at the Texas Children's Hospital, a sleep study at Kennedy Krieger Institute, the stem cells, organ expansion and communication, biomarkers at Duke, Boston Children's Hospitals, EEG biomarkers, citizen natural history study. We participate as Rare X and Simon Searchlight Registry. And all this means a lot of time, a lot of effort, you know, travel expenses. We also numerous hours on phone calls, questionnaires, but we understood as patients that this was very important to get to know what SINGAP1 is, and that's very important. So then we become, and we are inpatient patients, and that's how we change the path as SINGAP Research Fund, the path of SINGAP1 disease. And this is important also to address, because even though it is a recently diagnosed gene or found gene in 1998, but SINGAP Research Fund was formed in 2018, and in 4 years we have pushed into clinical trials readiness. We secured the ICD-10 code. We are leading rare disease advocacy group. We have put over 3.1 million in committees and grants. SINGAP1 right now is in preclinicals into public pipelines on ASOs, multiple approaches to small molecules, repurposing in progress. And we are working hard to get the biomarkers to find the best way to measure the impact of those therapies that we are trying to find, and also to have validated scales. So collaboration is definitely the key in order to go faster and still get it right. The first one is having a solid organization with entities in Canada and UK, in different countries, that also is going to make clinical trials possible as well in the different countries. European Union, and also how we can also secure legal charitable status, pool funds for researchers, and ensure governance and transparency. The grow the community is also very important, as one person was asking, because getting access to genetic testing for these rare diseases that are monogenetic and are epilepsy related to genetic, It is important to find, the diagnosis for the patients, have genetic testing available and accessible in different countries so we can have the patients ready. We can connect with families, we can support the families. Also, we can have that information, have the data, have the genetic data, the registries, and everything available. So it makes sense to the industry and the researchers to have all the information gathered. Also, professional community is very important, all the academic researchers, clinicians, biopharma industry, to make sense that the cause of and the effectiveness of this drug development have to have a sense on the return on the financial and all the financial return on this investment. Also, another important key element is to work together with other rare diseases organizations, is learning from those that are ahead of us, just like Dravet syndrome, Angelman syndrome, others that are already on clinical trials, and learn from their successes, also the challenges, and how we can make that improvement into our pipeline and also our preclinical process. And also, it's very important too, is to help other rare diseases that are also in the process of getting into trials or in this preclinical process. Government is also very important. It's important, to communicate well with the FDA, with the NIH, to have in place a very good public policy, legislation, regulatory framework, accessibility to drugs and treatments in the different countries. So that's very important. Collaboration is definitely the key element here. So in conclusion, clinical research is crucial to understand the disease pathophysiology. Before going to clinical trials, we need to understand what the disease is, what are the endpoints that we need, what are the outcomes that we are expecting from the trial, and what are the result of what we are expecting us. Patient engagement is understand our community needs to better set our goals, build trust, and focus product and treatment development. Also, getting the pathway to increase diversity into both research and trials. Having that patient engagement is going to also help us have that diversity that we are also aiming for, and making sure that those that are probably away from the cities where the research or trials are taking place can access to those trials, that they have the knowledge, they have the information, they have all the resources that they need in order to make the most effective and efficient decisions for their families. Know the other rare diseases, successes, and challenges. Once the clinical trials are into place, consent forms given to patients are must provide informative and clear information that addresses potential risk and purpose. The role of patients has truly changed in recent years, and this is very important. The voice now, our voice is a little bit harder. We need to collaborate since the beginning with pharma, with the industry, and with all the stakeholders, because we are an important piece of that development, of the drug development. Thank you so much.
Vicki, thank you so much. You made such incredible points in terms of the community lived experience role, the advocate role. Can you talk a little bit about, enthusiasms or fears that you've heard from your community and how they might approach providers or engage with any of those other stakeholders that you mentioned?
Well, definitely one of the biggest fears, especially when there are not as many patients, and one of them is definitely if we fail. That's one of the things. Like, what if the treatment fails? What if the outcomes are not the correct ones for the trials? Probably we have a poor selection of the endpoints. That's one of them too. And the biomarkers are not the right set, so we cannot measure those results on the trial. So that's one of the things. What are the side effects? What if the cost of the treatment? What if we are eligible or not? The age, and if the insurance is going to cover it too. So those are some of the fears on that. But the one point that is important to consider here is if we have a strong organization with patients that are engaged, it is important for us to have that in order to consider, how they can enroll to these clinical trials, how they can, maintain when they are in the clinical trials for a long time. So that's important for us because we are building that trust from the beginning, and we are giving the trust to them so they can also be engaged on either clinical research and the clinical trials at the moment.
Thank you. And there was a lot of questions that we addressed in the Q&A area about how people can get started in seeking out opportunities. And what was your experience? Did you start with your healthcare provider? You mentioned, you're not in that clinical trials phase yet, but even, in that preclinical trials phase that Dr. French spoke about, how did you engage or where did you start?
Well, I don't know this, okay, the first thing that you have to do when you have a diagnosis is reach out to the patient organizations, because those are the ones that are going to give you the first information. And those are the ones that that know a lot about your disease. Unfortunately, when we got the diagnosis for Amelia and we came to a neurologist, he didn't know anything about SYNGAP1. He didn't know what were the anti-seizure medications that work best for her. Probably short EEG wasn't the best recommendation. We need long EEG so we can have better tracking of the seizures that she was having at the time. So all of that information was given to me by the organization, but by the patient organization. So that's very important because you are also, it's not only about all the information on the clinical aspect, because sometimes it's very hard to understand when you don't have the clinical background, but also how could you make that information more understandable to the community. And what I'm working expressly on is about the Hispanic communities, how we can give meaningful information to the Hispanic community so they can make the best decision into their family's care. So that's what I think is important and why organizations are very important into the process of diagnosis.
Thank you very much. So I'd like to thank everyone for participating in the webinar this afternoon, for your engagement. I'd love to thank our speakers, Dr. French and Vicky Artiega, and also to our supporting sponsors, the Rare Epilepsy Network and Jazz Pharmaceuticals, for this offering. It's been a great session with a lot of great questions coming through in that chat. And the session is being recorded and will be made available on Epilepsy Foundation's social media and YouTube channels in the weeks ahead. Just wanted to mention one more opportunity before we wrap up for the day, which is that the National Walk to End Epilepsy is coming back to DC, Washington, DC on May 6th at Nationals Park. This is the first in-person walk in DC since before the pandemic, so in the last 3 years. If you'd like to learn more, please visit walktoendepilepsy.org/DC and get involved. We'd hope to see you there. Thank you again very much for your participation and look forward to connecting with you again soon. Take care.